Despite white eyes being recessive, throughout the entire experiment, there were more white eyed individuals than red eyed individuals. This ratio was relatively stable throughout the 5 weeks, with the exception of the second week, in which case the ratio was way closer to 50-50 between the two.
Based on the phenotypic ratio of the first week, we expected there to be 12.5 red-eyed individuals and 62.5 white-eyed individuals in the final week. With a degree of freedom of 1 and a significance value of 0.05, the critical value is 3.841. Since 0.024 is less than 3.841, there is not a significant difference between the calculated phenotypic frequencies of the first week and the last. However, we thought it was important to point out how the phenotypic frequency of the second week was significantly different from the first week, having a chi-square value of 14.49. Based on this data, our hypothesis is that the phenotypic frequencies would not significantly change was supported, with the exception of the second week. However, our hypothesis that the phenotypic ratio was not supported, as we hypothesized that there would be a larger amount of red-eyed individuals than wide-eyed.
Our results mean that the color of the flies' eyes have no impact on reproductive success, and thus, in a controlled environment, do not have an impact on fly fitness. We learned that in small populations, it's possible to receive phenotypes to have a higher frequency than dominant phenotypes, because when a trait does not affect fitness in any way, any changes in phenotypic frequency are due to chance. As a setup, we would repeat the experiment with new starting population sizes, such as 2 or 6 females for each phenotype.